In Chapter 4, we'll look at the structure of cell membranes, and we'll also look at the details of how these membranes function. As I've mentioned before, phospholipids are the most abundant molecules in cell membranes. Largely due to their interaction with water, they form a bilayer with the hydrophilic phosphate-containing segments facing the inner and the outer surfaces of the membranes. A variety of proteins are also present. These proteins give the membrane many of its special abilities. Complex glycolipids and glycoproteins are composed of polysaccharides attached to lipids or proteins. These molecules function as markers which identify individuals. The glycolipids and glycoproteins on my cells don't match exactly those on yours. Finally, cholesterol is abundant in animal cell membranes because it stabilizes the membrane. Plants don't need this cholesterol because they're supported by the cell wall. This drawing represents the structure of cell membranes. Because the phospholipids are not strongly bonded together, the membrane has somewhat of a fluid nature. Mosaic refers to the various other molecules embedded in the phospholipid bilayer. Notice how some proteins, shown in light blue, span the entire membrane, while others are just on the inner and outer surface. The green hexagons represent complex carbohydrates, which are attached to lipids or proteins. And then finally, in bright yellow, is cholesterol. Remember from lab, diffusion is the movement of any substance from an area of higher concentration and or higher pressure towards an area of lower concentration or lower pressure. The substance moving may pass through a membrane, but that's not required. In comparison, osmosis involves specifically the diffusion of water across a membrane. The direction of water movement is always from higher water concentration and or higher water pressure towards lower water concentration or pressure. A solvent is any liquid in which substances may be dissolved. In living organisms, the solvent is almost always water. A solute is the substance which is dissolved in the liquid. In a mixture of sugar and water, sugar is the solute and water is the solvent. A hypotonic solution contains a higher concentration of water and therefore a lower concentration of solute when compared to some other solution. Remember the demonstration in lab? The dialysis tubing containing molasses was placed in a hypotonic solution and water moved by osmosis into the tubing, filling it up. Another example involved the raw egg. Placed in a hypotonic solution, it gained water and its weight therefore also increased. Hypertonic solutions contain a lower concentration of water and therefore a higher concentration of solutes compared to some other solution. From lab, this was demonstrated by placing a dialysis tube containing water in molasses. Water moved out of the tubing and into the hypertonic molasses by osmosis. Iso means equal. If two solutions are isotonic, they have equal water concentrations. Because there's no concentration gradient, a cell placed in an isotonic solution will neither gain nor lose water by osmosis. In this diagram, the numbers represent the solute concentrations. Red is a membrane. Since the water concentration is greater in the blue solution in the beaker compared to the green solution in the apparatus, water moves by osmosis from the beaker into the apparatus. Plasmolysis is usually used to describe, in plant cells, the movement of the plasma membrane away from the cell wall, which is caused by the cell losing water by osmosis. Crenation is used instead of plasmolysis to include the loss of water from any animal cell. As a result, the cell shrivels up. Cytolysis is the opposite of crenation. An animal cell gains so much water by osmosis that the plasma membrane cannot withstand the pressure and the cell ruptures. This diagram illustrates plasmolysis, crenation, and cytolysis. The paint on the left shows an animal cell and a plant cell in an isotonic solution. There is no net movement of water in or out of these cells by osmosis. In the center pane, the cells have been placed in a hypotonic solution. Water enters the cell by osmosis, and the animal cell can rupture by cytolysis. Plant cells don't rupture because of the added strength supplied by the cell wall. Fresh carrots, apples, celery are crisped due to the resulting turgor pressure in cells surrounded by a hypotonic solution. Placed in a hypertonic solution, animal cells will crenate, and plant cells will experience plasmolysis. So far, we've looked at osmosis, which is a passive mechanism for water to move in or out of cells. Passive transport does not require the cell to do work, and the direction of movement is always down a concentration or pressure gradient. But larger molecules and charged ions typically cannot move by passive processes. Cells must be able to move these molecules across membranes, but that often requires active transport. The cells must do work to transport these items across cell membranes. Proteins play a central role in the transport of molecules across cell membranes. Let's review membrane proteins first. Peripheral proteins are found only on the inner or outer surfaces of membranes. They help to stabilize membranes and control the cell shape. Integral proteins reach all the way through the phospholipid bilayer. They perform a variety of different functions, including transport of molecules across the membrane. Let's look at 5 different integral proteins. Channel and carrier proteins are directly involved in the transport of materials across cell membranes. Receptor proteins enable the cell to respond to a variety of different signals. Some may involve transport. Recognition proteins enable cells to recognize one another. Finally, enzymatic proteins act as catalysts. They speed up the rate of reactions. Channel proteins are similar to open doorways through the membrane, which are accessible by only specific substances. For example, chlorine ions with their negative charge would be expected to get stuck to the polar phosphate-containing ends of the phospholipids. Instead, they can freely diffuse through channel proteins. Being diffusion, the direction of movement is dependent on the concentration gradient. The name carrier may be somewhat misleading. You might expect carrier proteins to be involved only in active transport. However, depending on the specific carrier protein, they may utilize active or passive transport. Both involve a specific molecule attaching to the carrier protein. This is followed by the protein drawing the molecule across the membrane as the carrier changes shape. In facilitated transport, the change in shape occurs without the addition of energy. Movement can be either into or out of the cell, and thus the net direction of movement will always be down a concentration gradient. Active transport also involves a specific molecule attaching to the carrier protein, but this time the change in shape of the carrier protein only occurs with the addition of energy supplied by the cell. Often the nucleotide ATP supplies that energy. Because the cell determines if and when to supply the energy, the direction of movement is independent of the concentration gradient. Sodium-potassium pumps are an example of active transport. Sodium-potassium pumps are vital in the transport of ions across the cell membranes during the transmission of information along nerve cells. Carrier and channel proteins cannot transport large items or large volumes of items. Endocytosis brings these items into the cytoplasm, while exocytosis moves items out of the cell. Phagocytosis is endocytosis in which the cells engulf large animals, including other cells, such as bacteria, or in this example, an old red blood cell by a white blood cell. The plasma membrane reaches out and around the item, eventually forming a vacuole containing that item, drawing it into the cytoplasm. Pinocytosis is another type of endocytosis. In this case, several molecules are drawn into the cytoplasm at the same time. This diagram emphasizes the role of molecules attaching to receptor proteins, which then trigger the transport. As you saw in the previous diagram, integral proteins can function as receptors, triggering a cell response. In addition to transport, other cell actions can result. For example, insulin binds to insulin receptors on cells, allowing them to respond to increasing blood sugar levels. Other integral proteins may function as enzymes. For example, the enzymes necessary to catalyze the reactions of aerobic respiration are found on the inner membranes of the mitochondria. Finally, integral proteins with specific polysaccharides attached function as markers which identify cells. For example, if some of my cells are introduced into your body, your immune system would recognize them as foreign because my glycolipids and glycoproteins do not match exactly your glycoproteins and glycolipids. These molecules are categorized as MHC, major histocompatibility complex markers. ABO blood type is one example of MHC markers. This completes Chapter 4. We're next going to skip ahead to Chapter 6 and start exploring metabolic pathways.